Good morning, everyone. It's nice to be with you. I'm Matt Peters, an associate professor in both our department and the Department of Neurology. Happy to see a lot of people I know in this room. It's great to be back in person. And for those of you on Zoom, thank you for joining as well. I picked specifically to do this presentation because he really exemplifies the hard work that goes into recovering after a traumatic brain injury. And not all of our patients are yet at a phase where they're ready to do that, but you've really jumped in, done everything that you need to have the recovery that he has. So thank you for being here. Another aspect of the case that we're going to talk a bit in the presentation about is that he had sleep disruption, which is something that we see very commonly after traumatic brain injury and ties in really nicely to the discussion we're going to have about the lymphatic system. I have no disclosures that are relevant to this presentation, and a little bit of background about why I chose this particular topic. Partially, the last time that I gave grand rounds, Peter Rabins reached out to me and said, "Hey, next time I want to hear an update on how you guys are thinking about traumatic brain injury." So giving grand rounds again today, I really wanted to honor that. And when we focus on traumatic brain injury and neurodegeneration, historically, there's been two trajectories following TBI: complete recovery after a milder TBI in the setting of days to weeks, and then static neurologic deficits after a more severe TBI. But since 1928, we've known that there's a third entity that exists in people with repetitive traumatic brain injury and progressive neurodegeneration, which is this term chronic traumatic encephalopathy. Uh, this was initially something really focused on in boxers, and then in more recent years, there's been a focus, especially in professional athletes like NFL players. And really, the group up at Boston University has been pushing a lot of this work along. And at this point, I think there's more that we don't know than that we know about this particular entity, and lots of research that needs to be done going forward. But another key point is now it's widely accepted by those of us in the field that there's an association between even a single traumatic brain injury. And it's important to note, in some individuals, not in everybody, but in some individuals, there's an association between even a mild injury and risk for future neurodegenerative illness. And that association is complex and complicated. These injuries vary by mechanism, by force, by directionality of the impact. There's certain brain regions that are more likely to be damaged, for example, those close to bony prominences in the skull. But you can really have damage in various parts of the brain from these types of injuries. Also, neurodegenerative pathologies have an insidious onset. So oftentimes, when you're starting to take care of somebody, they're 20 or 30 years after their injury, and you're really relying on retrospective self-report for the details of that injury. And a lot of those concerns come into play in the research realm as well. There's also different associations, including Alzheimer's disease, frontotemporal dementia, Parkinson's, and of course, CTE. So various associations that can go along with having these illnesses. And then in the true perspectives of psychiatry approach, these are people with their own personalities, behaviors, life circumstances, demographics, and those things almost certainly influence the way that people are presenting to us. So a little more background: since the lymphatic pathway was named in 2012, there have been 93 articles published on the connection to TBI. And this was a search that we performed in August of 2022. We put together a scoping review for Molecular Psychiatry, which is now in their hands and hopefully getting ready for publication in a few months. So that really drove the interest that they had in this topic, and an interest that I've had it in a while, really drove me to want to present it today. So the objectives of the talk today: I'd like us to understand the what and how of post-TBI brain cleanup, develop a basic understanding of the lymphatic system, examine how TBI disrupts this system, and then discuss some emerging trends in the literature. So when we think about the what of post-traumatic brain injury cleanup, what makes traumatic brain injury different than other forms of acquired brain injury is that you have an external force energy that is causing mechanical stress and damage in the brain. And what this results in is triggering of damage-associated molecular patterns, which go on to trigger a potent neuroinflammatory response. So this isn't a response in the setting of an infection, it's a response in the setting of the mechanical stress itself. And this external force that goes into the brain, parenchyma, the vasculature, the cerebrospinal fluid, you really have diffuse in different areas of the brain that can be impacted by it. And then in addition to this, we see an increase in extracellular amino acids, including glutamate, and this activates NMDA and AMPA receptors, and this leads to excitotoxicity. It seems to be a calcium influx dependent apoptosis, but we see that this can get triggered and then go on for an extended period of time. Additionally, we see both primary and secondary mitochondrial dysfunction, creating reactive oxygen species and further driving neuroinflammation. And we see, importantly, this blood-brain barrier disruption and cerebral vascular leakage. So something that's fairly well established at this point is when blood-based complement or cell-free hemoglobin gets into the CSF, it really can cause additional neuroinflammatory response. And that's happening in the setting of that blood-brain barrier being disrupted. Importantly to the talk right now, reactive astrocytes and microglial cells appear at the sites of injury, and there's conversations in the literature for what is more important, the astrocytes or the microglial cells, and at what time point are each of those two most important. And then lastly, we see this diffuse axonal injury, which is where you're going to get your accumulation of hyperphosphorylated tau or p-tau, amyloid precursor protein, and amyloid beta. And at least in some individuals, the release of these chemicals, even if it's just in one spot, has prion-like properties that allow it to propagate throughout the brain. So even though the damage may only be occurring in one location initially, there's this ability for transsynaptic spread of these molecules throughout the brain, just like you would see in the traditional prions that we learn about. So then how do things get cleaned up? So really, phagocytic clearance and drainage through lymph nodes is the mainstay of removing harmful substances from any organ system. And in the brain, we now know that that's essentially the same. So you have this clearance through microglial cells. There's targeted clearance across the blood-brain barrier. One example is that there's a low-density lipoprotein that can grab onto amyloid beta and get it across the blood-brain barrier. But that's a very specific targeted way of getting amyloid beta out of the brain. And then a lot of it's related to cerebrospinal fluid drainage. So the three main ways that CSF leaves the brain, and some of these are more recent discoveries, is through the superior sagittal sinus, which we all remember that one, and we'll talk about in a minute, the glymphatic system, which is going to be the primary focus of the presentation, and then also meningeal and nasal lymphatics. Interestingly, when CSF is moving through the brain, it is by bulk flow, partially from the choroid plexus secreting CSF, partially from arterial pulsation, which actually pushes some of this CSF along. And then they also know that during respiration, movement of CSF actually changes. So those changes in other elements of the way the body is functioning actually help to push and drive the CSF throughout the brain. So this is, I know it's going to be small for you guys, but this is showing the three different efflux pathways. So the first and most well recognized is this drainage through arachnoid granulations into the superior sagittal sinus and then out into the venous vasculature. I know, at least for me in medical school, that's the one that we focused on, and we didn't talk so much about these other newly discovered ones. The second is the glymphatic system. So as cerebral arteries dive into the brain, become peal arteries, and eventually turn into arterioles. And it's those arterioles that around them, they have a donut-shaped space that's called the Virchow-Robin space. It's been recognized for a long time. But CSF is able to flow down along the arteries following that space. So it actually flows down into that space. On one side, you have the peal membrane. On the other side, you have this astrocyte basement membrane. And it is actually able to use that to flow down into capillary beds. So that part of the glymphatic pathway was discovered in 2012, and we're going to come back to that in a second. There's another pathway that also exists along the arteries that actually has flow in the opposite direction of blood. The lymphatic system is following the flow of blood. This other pathway is going opposite the flow of blood. It's a perivascular space that only exists in arterial cells. So within one of the smooth muscle cell layers of arterial cells, there's a pathway that CSF can be pushed from that capillary bed and up along those arteries and then out into the nasal and meningeal lymphatics. And again, these are things that when I was in medical school, we didn't we didn't talk about. There was, a longstanding thought that the brain didn't have any type of lymphatic system, and now we know, in fact, it potentially has these two different pathways that are both potentially important. Okay, so now let's develop a basic understanding of the lymphatic pathway. So the name derives from the dependence on glial cells, in this case astrocytes, and its function as a lymphatic clearance pathway for interstitial solutes, and we'll talk about how it gets into that area. The seminal paper on this was published in 2012 by Ilf and colleagues, and if you look in the literature, these guys have done most of the publishing in this domain. There's a group at Hopkins run by Dr. Jessica Gill that's very interested in this work as well, but they're still thinking through some neuroimaging modalities that might allow them to do this in vivo in humans. But in that paper, which used a marine model, they were able to show that CSF enters the brain parenchyma along this perivascular space on the side of the arteries. It mixes with interstitial fluid, and then that CSF ISF mixture drains out along paravenous drainage pathways. And an important thing that comes up in the literature over and over again is that there are channels called aquaporin 4 channels that are a mechanism for water to shift between the CSF and the ISF. And these water channels, they primarily focus on astrocytic end feet. There's a lot of polarization to that region. And it's thought that they're absolutely essential for the efficiency and effectiveness of the system. So another thing that this same group showed, they did it in two different papers. One was in the seminal paper, one was in a paper focused on traumatic brain injury, is that when you disrupt this system, there is an accumulation of soluble amyloid beta and hyperphosphorylated tau. So that's how this all starts linking back to the possibility of neurodegeneration, really regardless of what neurodegenerative disease you're talking about. Okay, so this is another photo that we put together. So you're seeing that para-arterial CSF influx. You have those astrocytic end feet that it gets into the interstitial fluid. There's mixing that occurs. There's convective forces that push it. And then eventually you have this paravenous efflux at the other end. When we talk about the astrocytic end feet, not only do they have these aquaporin 4 channels, but they also have little spaces in between the end feet themselves. So that is one mechanism. Those clefts is one mechanism that water and other solutes can be exchanged, and that's in a size and molecular pattern dependent manner that it does that. But then these aquaporin 4 channels, for this drainage, this idea of drainage into the lymphatic system, appear to be basically essential for the functioning. Two important points before I move off of this slide. One is that this lymphatic system essentially shuts down during periods of neuronal activation. So when your brain is firing on all cylinders, it's one way that extracellular calcium gets brought back into the cell and you actually shrink the interstitial fluid. So during neuronal activation, lymphatic activity goes way down, and during sleep, it goes way up. The interstitial space expands by about 60%, and there's a growing consensus that potentially this is the reason why we sleep, that this pathway gets activated, it's taking out the garbage at the end of the day. And if you have sleep disruption in the setting of a neurodegenerative condition, all of those things that have built up are not able to get out of the brain. So how can a traumatic brain injury disrupt the lymphatic system? So like any growing field, the in vivo human research right now is basically lacking. It's really focused right now on how do we develop in vivo neuroimaging modalities of flow through this system. And what's important is that a static picture doesn't teach you very much. You actually want a picture of the lymphatic system functioning and flowing. So there's a number of imaging modalities that have started to be developed. Some of them involve actual gadolinium injection into the CSF. So injecting it directly into the fluid and using that as a way of watching this flow into the brain parenchyma, also using contrast enhanced FLAIR, and then this ultra fast MR encephalography. There's not enough to spend a lot of time on this topic, but for our neuroimaging colleagues and anybody else that's interested, the PubMed IDs at the bottom is the existing literature that describes these things. And then the rest of the literature is really from marine data, so either rat or mice research. Currently, there's 6 studies on the influence of TBI on lymphatic system disruption. And just briefly, 2 of them actually use that injection of gadolinium, but again, using a mouse model. One of them is looking at morphological and histological change, so postmortem changes, focus specifically on the glymphatic vascular space and astrocytic end feet morphology. And then there's quite a lot of studies, 3 of them that have looked at astrocytic end foot, aquaporin 4 expression and polarization, and looking at that as a way that traumatic brain injury disrupts this pathway. So I'm just going to put a summary up of the findings from those studies, and then I'm going to dive more deeply into just one of them to give you an idea of what this research looks like. So to summarize the existing data, it's pretty clear that a traumatic brain injury decreases flow through the lymphatic vascular space, that there's a loss of those aquaporin 4 channels on the astrocytic end feet. So that doesn't mean that they're not there. They're just no longer polarized to the end foot. And if they're not at that sort of CSF ISF interface, then you're not going to be getting water from the CSF like you're wanting to. And so the flow doesn't happen the way that it's supposed to, even if those aquaporin 4 channels are still there. And some of this appears to be driven by the astrogliosis that can happen after you have a neuroinflammatory response. So as the astrocytes change their shape, it appears like they are less likely to have that polarization along, whether it's the CSF ISF interface or CSF blood interface. And then if you look at aquaporin 4 knockout, it appears to be a double-edged sword. So interestingly, if in a model of severe traumatic brain injury, if you have aquaporin 4 knockout, it actually prevents cerebral edema. So in a more severe injury, you're preventing that flow of water, which can lead to cerebral edema in the brain, which is an interesting and potentially good thing. However, if you have disruption of those aquaporin 4 channels, then as time goes on, you're not able to clear out those different proteins that get deposited abnormally. And so in the long run, aquaporin 4 knockout, especially in more mild injury, results in exacerbated protein aggregation in the long time, including that hyperphosphorylated tau and amyloid beta. Additionally, and I just said this, decreased proteins in TBI-related neurodegeneration, so amyloid beta and p-tau. And then there's only one study, but it's really interesting. So we've known for a long time that when you think about blast injury, even if somebody receives a blast to, let's say, their torso, they can still end up with damage to their brain. And one of the reasons that this is thought to occur is the blast actually gets absorbed into the vasculature, and that sends oscillations up into the brain that can cause what very much looks like a traumatic brain injury, even if the head itself wasn't technically part of the blast. And most of this research, I mean, this is not human research. Most of this is going to come out of, mouse modeling where they're giving a blast to a different part of the body and still seeing that increase in proteins that would go along with traumatic brain injury. And so when they've done studies using blast as exposed to an impact traumatic brain injury, there actually appears to be more diffuse damage both on the side of the blast injury and on the opposite side. So just looking at one paper, this is a study by Ilf and colleagues in 2014. They used a mild to moderate closed head injury mouse model, and they used both aquaporin 4 normal mice and aquaporin 4 knockout mice. And what they found was that the normal mice exhibited a 60% decrease in lymphatic pathway functioning that persisted for up to 28 days post-injury. And if I can show a picture of this, so on the left-hand side, you see the sham mouse that didn't receive an injury. The right-hand side, labeled E, you see 7-day post-injury imaging. And this is a tracer that is meant to go into the cortex and light up those paravascular spaces. And in the mice with the traumatic brain injury, both on the ipsilateral and the contralateral side, you see this real knockdown in how deep the tracer was able to penetrate. So probably for you guys, if you look at D versus F, D is the sham, F is the ipsilateral side and head injury, there's a pretty clear decrease in the ability of the tracer to transfuse along those para-arterial pathways. And although they don't highlight it and bring it up in this image, the same in this particular study was true of the contralateral side. Then they did something similar in aquaporin 4 knockout mice, and they showed further impairment in lymphatic function. So We already had the impairment. If you knock out aquaporin 4, the impairment looks even more severe. And importantly, doing this particular change where you're getting rid of the aquaporin 4, you see an exacerbation of p-tau neurofibrillary tangles, axonal degeneration, and then decreased neurocognitive function by behavioral testing. And this is just a graph or a chart looking at hyperphosphorylated tau immunoreactivity. So higher on the y-axis is going to be more accumulation of hyperphosphorylated tau. The figure is a little hard to understand, or at least it was for me, but on the right-hand side, wild type, you're looking at contralateral and ipsilateral side. And then when you look at the aquaporin 4 knockout, you're really seeing this deposition in both the cerebral cortex and the striatum of hyperphosphorylated tau. And interestingly, in this, you're seeing that hyperphosphorylated tau is only on the side of the injury. So even though we saw in the other image that the lymphatic pathway functioning was decreased on both sides, here we're only seeing the deposition or the increase of the hyperphosphorylated tau on one side. And to me, the easy explanation for that would be that that is the area where the actual diffuse axonal injury or initial release of hyperphosphorylated tau occurred to begin with. And so that's where you're seeing that deposition. So even though the lymphatic system is disrupted on the other side, you don't have this sort of massive damage that occurred from the impact itself. Okay, and then wrapping things up, I just wanted to discuss a few emerging trends in the literature. So there's going to be 3 trends. One is related to sleep, one is related to biomarkers, and then the third is related to development of therapeutics. So we know very clearly that sleep disruption is very common after traumatic brain injury. It occurs in 30 to 70% of individuals. There's pretty good data suggesting that in more mild cases of head injury, you're actually more likely to see this sleep disruption, but that hasn't always been consistent in the literature. And then those with sleep disturbances after head injury have slower recovery trajectories and more post-concussive symptoms. This is just a bit of data from a Venture Discovery Fund award that we got, where we were taking sleep devices, including the sleep profiler, and seeing if we could prove the feasibility of using it directly within our community psychiatry clinic, as well as within the Memory and Alzheimer's Treatment Center. And he did the study for us, so he took this equipment home. And I'm not going to run through all the numbers, but you're essentially seeing, if you look at the column all the way on the left, you're seeing, 47%, 53%, 40%, 73%. So a lot of abnormality, no matter what sleep parameter you're looking at, whether it's sleep time, sleep efficiency, sleep latency, or wake after sleep onset. I didn't put normalization on here because that depends by age, but just take my word for it that there was a number of abnormal sleep parameters when we did the sleep profiler, and then something similar showed up when we're looking at the actual sleep architecture. So really a lot of abnormalities in the sleep of these individuals. And if you look at all the parameters that we assessed, the median number of abnormal was 5, with a minimum of 1 and a maximum of 8. Of course, happy to dive more into this data at another time, but the main takeaway is, just like is taught in the literature, we really did find a lot of sleep disturbances in the patients that were taken care of. And then this is from a study by Z et al. In 2013. And they actually wanted to look at, in sleep, what happens to the interstitial space. And we already talked about this a little bit. They found that it expands by up to 60%. They found that the functioning of the lymphatic system is at its peak during non-REM slow wave sleep. And the thought is that there's these normal oscillations that happen in blood volume during that phase of sleep. And if the blood volume is oscillating or fluctuating, remember that CSF is sitting right next to it. And so it kind of pushes it along in the same way. And so you're seeing the most lymphatic pathway functioning in that non-REM slow wave sleep. CSF production also increases during sleep, which helps to drive things forward. And then just from this study specifically, they looked at how amyloid beta is cleared, and it's cleared at a faster rate during sleep, and also interestingly in an anesthetized state. So in this particular chart, you're seeing the rate constant. So how fast are things being cleared? How fast is this amyloid beta being cleared? In the awake state versus the sleep state, you see this significant jump. So clearing a lot more amyloid beta during sleep. And then interestingly, same thing if you just anesthetize the mouse. So this is more than just circadian rhythm functioning. There is something too, if you can put at least the mouse asleep, you see this increase in functioning. The second emerging trend is related to biomarkers. So for any of you that know the literature, it's been very challenging to develop biomarkers for traumatic brain injury, especially ones that give you any idea of prognosis. And this was a study by Plog et al. In 2015. They used a mild to moderate, again, mouse model, and there was almost a complete suppression of clearance of TBI-related biomarkers. So looking at these graphs, you have S100B, glial fibrillary acidic protein, and neuron-specific enolase, 3 markers that over and over again get looked at in the literature as potentially being diagnostic in people with traumatic brain injury. The control group, those are just mice living their life. The TBI group obviously received a TBI. And so if you look at between those two, you see this dramatic increase in the levels of all 3 of those particular biomarkers. Now what they started to do is they started to do manipulation. So the first 2, they actually, one of them was giving acetazolamide, one of them was a cisterna magna cisternotomy. Both of those cut down on the forward pressure of CSF through those pathways. And then they also did knockout of aquaporin 4, and they did sleep deprivation. And even though it's small for you guys, if you look at all of them, you see this dramatic decrease in the biomarker levels. So obviously that's pretty problematic if you're trying to assess for a traumatic brain injury, and it's pretty clear if the glymphatic system is disrupted, you don't get the elevation in biomarkers that you would expect. So a lot of the biomarker folks are now trying to figure out how do you add in a measure of glymphatic pathway functioning when you're assessing traumatic brain injury biomarkers. And lastly, like any good topic, there's already therapeutics that are starting to be discussed. There's some thought that you could use stem cells to inhibit reactive astrogliosis. And as I said earlier, that leads to this depolarization of aquaporin 4. So trying to get that polarization to stay. Use of anesthetics after head injury. So if that mouse model was correct, that you can get increased flow through the glymphatic pathway by putting a mouse to sleep, could you do the same thing in a human and increase the clearance through the glymphatic pathway? And then the osteopathic literature has really picked up on this. There's a number of manipulations that they believe increase lymphatic pathway drainage, and they're now trying to translate this into lymphatic pathway drainage. And there was one study that showed fairly significant decreases in markers of increased intracranial pressure after a head injury. Please note this is only in 11 patients, and this would obviously need to be replicated, but looking at both intracranial pressure, and a pupillary index and an optic nerve sheath index, they saw decreases in all of those, suggesting that they were actually decreasing this increased intracranial pressure by doing these manipulations. So just some concluding remarks. This is a relatively new field. It's grown exponentially since the seminal paper in 2012. There are similar growing literature bases for normal, quote unquote, aging, Alzheimer's disease, and Parkinson's disease. There's a current reliance on these marine models. There's a need for developing biomarkers to assess lymphatic system functioning before we can do in vivo studies in humans. And really the literature is still at the stage of proving disruption of lymphatic system flow and evidence of protein accumulation and deposition. So although I sought out to look specifically at its role in TBI-related neurodegeneration, we're still just getting to that point that that's something that different teams are studying. And then as we just concluded with, emerging topics include influence of sleep disruption, considerations for biomarker development, and then novel therapeutics. So thank you guys for listening. Any questions?
Thank you, Dr. Peters. Great talk. Fascinating topic, the glymphatic system. Maybe this is, I'm sure this is quite simplistic, but it seems like an obvious thing for people to look at that is the question of would more aggressively treating sleep disruption in people with TBI potentially improve the clinical picture, presumed by virtue of somehow improving lymphatic drainage.
Yeah, and so we actually put a grant in to the Precision Medicine Alzheimer's Disease Center. They had a request for applications. It didn't get funded, but the idea is still sitting out there, hopefully for one of the donors to pick it up. But looking at really exactly that, like, is this potentially a treatable condition that would improve outcomes? Is this a subset of individuals that treating them you would see an improvement, which is really what precision medicine is all about. How can you identify that subgroup, get them available treatments, which we know exist, and see how that influences outcomes. So it was one of the motivations for doing the study in the clinic to begin with, is we know that this causes serious disruption after traumatic brain injury, and yet it very often doesn't get assessed in the clinics. You get referred to a sleep specialist, and then you have 6 months to a year wait, just like you have to see us, unfortunately.
Right.
So it's patients have a multi-year process before they even get the workup done.
Right. Exactly. Other folks have questions? Go ahead. Dr. Wilson.
Mm-hmm. Yeah, I might maybe I phrased it wrong.
Go ahead and repeat the question.
Yeah, so and you make sure that I get it right. So the question was, I discussed hyperphosphorylated tau clearance being decreased after a traumatic brain injury, potentially because of this lymphatic system disruption. The biomarker clearance also goes down. So if I didn't say that correctly, my apologies, but it's the same direction. It's, and maybe it was the chart that was confusing, but after a head injury, you get these spikes in the periphery, in the blood of those biomarker levels. So the spike you're actually seeing out in the blood when you're talking about biomarkers. So if the level of biomarkers out in the blood is less, the idea would be that more of it is still in the CSF. So that's a really important clarification point, actually, now that I'm saying it. So yeah, one is you're looking at out in the blood, and then in the CSF, the hypothesis would be phosphorylated tau goes up, S100B, neuron-specific enolase, GFAP all go up as well. They just can't get out for you to actually see it in the blood. So like the cool follow-up study to that one would be to see, okay, it goes down in the blood. What are you seeing in the CSF? That would be a nice study for them to do next. I don't think that was part of the manuscript, but that's a great clarification. Thank you.
Other folks? Yeah, go ahead.
Hey, Michael.
So you mentioned that the animal model, it was the NREM sleep that they were looking at, like slow wave sleep. But if I read your slide, right, it looked like in your study that abnormality were in like REM.
Yeah.
And I guess I'm wondering, like, what's happening in the lymphatic system during REM sleep, and if there's that decreased latency that REM once it's like a wakefulness issue.
Yeah, no, great question. So no matter what,
why don't you repeat the question?
Yeah. So Michael specifically asked about the studies that they've done show that within mouse models during non-REM sleep is where you have the greatest functioning of the lymphatic pathway. When we've done this work within TBI populations, that's not necessarily the indicator that's disrupted. Michael, the increase is for all elements of sleep. So lymphatic system functioning goes up during all elements of sleep, but goes up the most during non-REM sleep. And the only thing that I would say about our study is it's only 30 people. And so I would lean more towards that's what they've shown in mouse models. It has not been shown in a human population or a human sample. And I'd have to actually look at the literature to see, do the findings from our 30-person study line up with what the sleep architecture disruptions look like in TBI in general. I don't know that off the top of my head.
Other thoughts? Let me come back to the treatment of sleep issue, Matt, because you were talking about putting in a grant to do that empirically, or for clinicians right now who are taking care of patients with TBI, would you recommend being more aggressive in the treatment of sleep than a clinician might ordinarily be? Is there reason enough to think that it might be useful that people should be more aggressive? And if so, how?
Yeah.
What would that look like?
Yeah, in both the TBI population, really any neuropsychiatric illness where you're having some sort of deposition as the driving cause of the psychiatric presentation or of the sleep disruption, it's oftentimes one of the very first things that we do in memory clinic or in our community psychiatry clinic is do a sleep assessment and determine, should we send this person home with equipment? So in my eyes, the ideal setup would be that we as psychiatrists understand enough about sending that equipment home, not necessarily interpreting it, because the sleep medicine specialists, they can bill for interpreting it. They're more than happy to help us with that. But having the ability to send an assessment home with the person, ideally one that assesses for sleep apnea, which our device didn't. And then if there is a finding there, then you got to get them referred over to sleep medicine. Now, there's still going to be a hang up for, okay, you've identified it, but that doesn't necessarily get them seen sooner. So we haven't really thought of a great mechanism for us referring directly to like an inpatient sleep lab out of our clinic, but I don't see why that couldn't be possible. So ideally, something at home that the patients can do easily, simply gives you a picture. And if there's a finding there, whether it's sleep apnea or other sleep disruption, get them over for like an official sleep evaluation. And again, the sleep specialists work with us a lot, so they're excited and happy to help us. They're just under the same wait list that we are.
Great, thanks. Other folks? Yeah, go ahead, Dr. Burt.
Is there any data on the new, like the dual orexin receptor antagonist in the TBI population?
Yeah, great question. I haven't seen anything come out. I mean, it potentially would be really interesting. I've tried to prescribe it once and it wasn't covered by insurance. So even when I tried to use it, I didn't actually get to see how it would impact the individual.
This is suvorexant. Is that the drug you're talking about?
Yeah. It wasn't approved. It wasn't even close to being approved, I think. But no, I mean, that's just going to keep coming out. We still have a problem, and this maybe will be my last comment. We still have a big problem in the traumatic brain injury research field in general with how do you properly identify the individual that's had an injury. I mean, mostly now it's done based on subjective self-report. His skull to be open, that's an obvious head injury. But 90% of the people we take care of, they've had a mild traumatic brain injury. Clearly something is different about them after that injury. But proving and choosing the right people to be within a clinical study has proven to be very difficult. One great thing that Greg Pontone taught me about Parkinson's research is he likes it because you can see that motor change and you have a pretty good sense of, this is a person with Parkinson's disease that I'm going to put in my study. In traumatic brain injury studies, we have a really hard time making sure that we've identified the right people.
Great. Yeah, go ahead, Dr. Onaya.
Yeah, sure. So two important associations. One,
You want to repeat the question?
Yeah, so the question is there any association between severity of sleep issues and the severity of traumatic brain injury? There's two parts to that answer. One is when it comes to subjective sleep complaints, way more common in mild traumatic brain injury, actually. When it comes to things like central obstructive sleep apnea, which we see as as opposed to peripheral obstructive sleep apnea, you end up seeing more of that in individuals with severe head injuries. So some of the sleep apnea type stuff is more likely in those more severe cases. But I would say the majority of patients in our clinic actually have sleep disturbances. It's one of the primary things we focus on.
Thanks, Dr. Bryan, for your presentation. And thank you, Dr. Peters, for a very illuminating.